machine learning algorithms in the prediction of blood-brain barrier permeability. The development of drugs targeted to the central nervous system is a very long process, up to 16 years, and has a very low success rate. This is due to the extraordinary complexity of the brain, the high degree of side effects, and most importantly, the presence of a blood-brain barrier. This barrier is a major problem because it acts as an almost impenetrable, highly selective membrane to which 95% of molecules show poor or no permeability. Drug design is thus very difficult, requiring a large number of molecules. Therefore, it is of paramount importance the development of prediction and pre-screening tools for large chemical databases. The aim of this work is to test different machine learning approaches in both balanced and imbalanced datasets. Four main conceptual tools were used in this thesis. Molecular descriptors, these are the sort of representations used to capture structural and physicochemical properties. Making use of fingerprints, molecular similarities were computed using all these distance measures seen here, so one similarity matrix per distance measure. Principal coordinates analysis was also used. It is analogous to PCA, but it takes a dissimilarity matrix as input. It basically converts high-dimensional data into a lower-dimensional vector of coordinates for each molecule. Three machine learning algorithms were chosen: k-nearest neighbors, a neural network, and random forests. Identical pipelines were built for classification with KNN and neural nets. They used an imbalanced dataset 1, from which fingerprints representations were acquired and dissimilarity matrices computed. For each dissimilarity matrix, a PCOA outputs a vector of coordinates. Only then, molecular weight and log-p variables were added and fed to the ML algorithms. Different dimensions of PCOA were also tested, as well as a variable number of neighbors for KNN or neurons in case of the neural net. Results for KNN and neural nets. First, it was analyzed the impact of molecular weight and log-p, which is a measure of solubility, because traditionally these are two important factors that may explain why certain molecules cross the barrier. However, alone, they don't seem to be enough for this classification, with below 5 values. Regarding the number of dimensions from PCOA, in general, the best results require a higher number of dimensions. And besides the number of dimensions, k-nearest neighbors depend obviously on the number of neighbors, although here there's no conclusions because it seems to be dependent on the distance used. On the other hand, in neural nets, an increase in the number of neurons yielded better results. The third algorithm, random forests. Here, dataset 2 with all molecular descriptors was used, not just fingerprints, and for clarity, no matrices or PCOA were computed. The main difference from dataset 1 is the oversampling of data points. Given that class priors are highly unequal, meaning the non-crossing molecules should be 95% of samples, it was decided to do a replication of the non-crossing molecules. One advantage of random forests is that it allows the analysis of individual variables by looking at their impact in the decrease of accuracy. The author mentions three particular descriptors. One, the number of bonds in molecular rings. It seems that below a certain threshold, there is a high probability of being classified as crossing molecule. Second, also interestingly, is the fact that the maximum of average weight is very similar in both classes, where it was expected a lower weight on crossing molecules. And finally, and most importantly, random forests choose the number of oxygens with one single bond as the most important variable to distinguish between crossing and non-crossing. Discussing the KNN, the comparison between KNN and neural nets, the overall accuracy and phi were similar. The difference lies in KNN predicting crossing compounds, whereas the neural net predicts non-crossing molecules better, and is also better to handle the imbalanced data sets. And also KNN requires a lower number of dimensions, which is advantageous. The variables molecular weight and log-p do not bring an improvement. The search for better results led to random forests implementation. It shows a higher phi value, gets a higher predictive capability for the crossing class, and the ability to identify the most important molecular descriptors for this particular classification. In conclusion, of the three statistical learning methods tested, random forests appear to give the higher prediction accuracy and phi values, mainly for molecules able to cross the barrier. The use of structural features, as for example fingerprints, seems to be a good approach and quite enough for correct classification, and the use of prior probabilities of both classes on random forests appears to increase accuracy and phi values. At last, the use of random forests for this problem is not new, but this approach brings novelty by the used variables and their number, the oversampling, and the application of prior probabilities. Thank you.